Welcome to New York Bio's Virtual Breakfast Series, a digital program started in 2020 featuring fireside chats with leaders from across the healthcare spectrum. This week's episode brings us Chris Marshall, the founder and CEO of Calder Biosciences.
All right. Good morning, everybody. I have 9:01. We'll get going. My name is Jennifer Hawks-Bland, and I'm the CEO at New York Bio. We're thrilled you're here with us for another edition of our virtual breakfast series. Hopefully everyone has their coffee. Chris apparently is out, but Derek and I are still fully caffeinated. So we will be peppering him with questions. We're thrilled that you're joining us today. And thank you to Moses and Singer, our partner. And Moses and Singer has not only partnered with us on our breakfast series this month, but also a partner at Moses and Singer, Linda Malik, is co-chairing our Equity, Diversity, and Inclusion Task Force. So we're really thankful to their engagement across a whole spectrum of New York Bio activities. And with that, as always, my housekeeping notes for you. Please put your questions for Chris in the Q&A or in the chat, and Derek and I will get to them as we go through our conversation. We're thrilled to have Chris Marshall from Calder Biosciences here with us today, and I am going to kick it over to Derek to do a more thorough introduction.
All right, good morning, and Chris, thank you so much for being here. So I'm excited for all of these, but I'm particularly excited for this one. So as some people know, I started a networking group for biomedical entrepreneurs in New York City a little over a decade ago. And I filtered all the way back to the beginning. And as it turns out that Chris, you and I met actually just a little bit before that first meeting through a good friend of ours named Arthur Tinklenberg. So I've been privy to kind of your entire entrepreneurial story over the last decade. And really it's just a great one. And I'm super excited to have you here this morning. So thank you so much for being with us.
Thank you for having me.
All right, so if we dig in, so you are what I would call almost the prototypical New York City entrepreneur. Came out of Rockefeller and eventually started multiple companies in the bioscience space. So can we wind the clock way back to graduate school and talk a little bit about where you came from scientifically and how you got to your first company?
Sure. I mean, when I was at Rockefeller, I was working in the Hidesaburo Hanafusa lab, and we were focused on tyrosine kinases and cellular signaling and all that stuff. And so even back then, I was focused on tyrosines, which so has been really that has gone through my entire career, these tyrosine side chains. So I really had a wonderful time in graduate school. And then when graduate school was over, I just, I looked back and I was like, is this how I want to keep going? Both of my parents were academics. So I really took a look and just figured sort of like, I just felt sort of like I needed to do something different. And so then I applied to a bunch of management consulting firms and got into Oliver Wyman. And so I started focusing on the financial services industry, which was quite a jump. When you're coming from life sciences into financial services, that's you really don't know where you're at for at least for a while, there. And so we were doing risk management and general strategies from a risk perspective. It was a lot of risk. So when you think about risk, I mean, that's just what entrepreneurship is really all about. So I was at Oliver Wyman for a while. And then, I guess this was in the late 90s. And a good friend of mine was a lawyer at Morrison Foerster, and he suggested that I start a company, because he was advising all these people who were starting companies, and he just figured sort of like, start a company.
What year was this?
This is probably 98.
Yep, that sounds about right.
Yeah. And so then he put me together with some clients of his who were sort of like, had already started a company. So we started talking, and we sort of got working, and one thing led to another, and eventually sort of like I pulled some friends together, and we started Avatar, and focused on the idea was first to get to revenue before we needed regulatory approval, right? We wanted to get around. We wanted to do biotech, but we didn't want to have to go after regulatory approval because that's so expensive.
Right.
So we wanted to get revenue positive as quickly as possible. So the idea there was to go after industrial enzymes. And first it was industrial enzymes, then it was protein chips, and then sort of like, the technology kept on working better and better and better. And we were just like, sorry, hold on, wait a minute. Maybe we ought to be going after therapeutics after all. But the company really was an industrial enzymes company. And so one thing led to another, and we started, eventually we ran out of cash. It's really sort of like just what happened there. And then I realized the importance of patents. And the cost of patents is really sort of like, one of the main drivers there. So I sat down and studied and got through the patent bar, so that I could file my own patents or become the ultimate nightmare of a client. Really, that's the ultimate outcome here.
So wait a minute, you looked around and said patents are super expensive, so you decided to just study and become your own patent agent.
Yes.
Like that's one way of reducing the cost that generally does it. It's not the tried and true way to reduce your patent costs.
Yeah, well, I mean, that worked for me.
Yeah.
And then I actually filed some of those patents in those early years, and those are not the best patents. So there is more to patent prosecution than just getting through the patent bar. So I was licensed and was able to file my own patents and had learned how to do it and all that stuff. You don't actually even need to be licensed to file your own patents. Anybody can file their own patents. But it's really good to work with people who have experience. So that's been my experience there. So anyway, then I got involved with some friends. I was doing a whole bunch of consulting work to stay, just to stay abroad, alive, and then I got involved with another set of scientists who were coming out of Rockefeller and Howard Hughes, and so we licensed technology there and started Ophidian. Got sort of like got grant funding going, got some patents filed, sort of like did that whole thing, and then in the process of negotiating the Series A, I was asked to resign, sort of like as the CEO, because the investors wanted to bring their own investment, their own management team in, which I was very happy to do. So then I went back and still had this technology from Avatar with the tyrosines, and I just figured sort of like, I never let the patent prosecution go. I just figured somehow we got to be able to use this. But at Ophidian, I was working with my friends Niva and Jahan, and Jahan at the time was working at IAVI, and so then she was able to figure out sort of like, why don't you try to apply your technology to HIV vaccines? And so then I applied with IAVI to the Gates Foundation. And then, so the Innovation Fund was IAVI money and Gates Foundation money together. And that's how we then got started in vaccines. And then sort of like, we kept working in vaccines, and eventually we sort of like rebranded and called it Calder now. So
right.
As we moved away from industrial enzymes, we kind of needed to be a new company. So we're now calling it Calder.
Right. I mean, so now you're a vaccines focused company, and where are you in terms of development? What do you have kind of on the table, and what are you working on?
Right. So so we started in HIV, and HIV is a really brutal space, or at least it was. I think today it might actually be a different story. But at the time, it was really not an easy place for a startup company to get funding, or to just do what you need to do in order to make money. So we got to proof of principle. We really did some very good work in HIV, and I'd like to go back to it. But to be more commercially focused, we started focusing on RSV and universal flu. So those were the two opportunities that we were able to identify from, we live within IAVI here now. We're in an Army terminal. So we live within their design and development lab. We've been renting from them for a long time now. So from this perspective, we were able to identify the universal flu vaccine and the RSV vaccine opportunity as truly commercial opportunities to go after. And so those are the two that we've gone after. The influenza project is much more complicated from a technical perspective. And so we've made a lot more progress in RSV. And so that's where we're now the furthest. And so now we're pushing our RSV vaccine. So we've basically gone from applying the technology to testing it in mice, to testing it in cotton rats, to applying the technology again, and sort of like very iterative approach there, because the technology is a process technology, and so making it over and over and over again just makes it better and better and better, and so now we're approaching an order of magnitude better than what we started with, so our vaccine is really looking very, very good now. So now we're going into manufacturing. We're making cell lines and doing the process design and development, and then we hope to start clinical development, as soon as we're through manufacturing, tox, and IND file.
That's fantastic. So
I was going to say, where are you manufacturing? Are you doing it there, or have you contracted that out?
Right. So we're working with a company called Abzena. So they're doing cell line development for us, and they'll also be doing the manufacturing in their San Diego facility. Right now, we're sort of like still sorting out sort of like, what columns are we going to use for capture? What's the first step? What's the second step? How are we going to get through viral clearance? We're doing everything in CHO cells, so everything is CHO specific. So there's just a lot of things that we can address in our lab. So we're doing that, so that when it comes time for technology transfer, that it will be ready to go, and then we can really push through this very quickly. So we're now at a place where we can we have all of the risky steps are addressed, and we can get through. We hope to be able to get to an engineering run within about 6 months of technology transfer. So we're really at a great place right now.
So just for the edification of the audience, so RSV is respiratory syncytial virus.
All right.
Right. Largely affects anyone with kids knows what RSV is, because you show up to the doctor and your kid has a snotty nose and they say, that's probably RSV. But I think the main two things that people look at for RSV are infants and the elderly. Correct? Are you kind of focusing on either patient population?
So no, we want to go after both,
okay.
And then there still is sort of like this possibility someday of trying to go after direct pediatric vaccination. So there's children that are right now being addressed by maternal vaccination.
Yeah.
And there may be direct pediatric vaccination, school age, children eventually, or whatever, and then the elderly. And there is also, at some point, you could consider a co-formulation of a flu vaccine with an RSV.
Right. And again, when you say universal flu vaccine, it's not a seasonal flu. You wouldn't have to get it every year. Effectively, this it targets, if I remember correctly, the stem of the virus instead of the head, and the stem doesn't mutate. Correct. So you get it once and you're done. No more flu vaccines.
Exactly. So the idea there is that the surface protein, which is the fusion protein of the influenza virus, looks like a mushroom. So if you imagine we've got a stalk and a head, and the head is immunodominant and variable, and the stalk is the business end of the molecule, as I like to call it, and it's completely conserved. if you can engineer this thing in such a way that it maintains that shape, right? Because vaccines have to have that same three-dimensional shape.
Right.
But, you know, that's that whole thing, you know, so like that antibodies recognize three-dimensional shapes and surfaces and all that. And so if the vaccine doesn't present the same shape as the pathogen does, then the antibodies raised to the vaccine won't recognize the pathogen.
Right.
So you need that cross-reactivity. So we need to maintain that shape and not this. We don't want to present this shape. We want to present the stalk in its shape. So so what we do is we express the whole protein so we get the right conformation. Then we lock up the stalk with our dityrosine crosslinks. That's the technology. So targeted dityrosine crosslinks. So we lock it up and make sure that it holds that shape. And then we proteolytic remove the head and purify it away and stuff. So that's a, you know, when it boils right down to it, that's a very, very complicated process that I just described there. You're just getting dityrosine bonds to form in a process, you know, when you're in manufacturing is not easy. So we've spent many, many years trying to get that to work. And that now works very well and very efficiently. But now adding on top of that we need to have a proteolytic cleavage process and remove the head. It's complicated, but we'll get there. And it'll be a great headless molecule. And headless is really a wonderful way to protect against flu.
not good for humans.
Nope.
Protecting against the flu virus.
Yeah. We actually have a good question here, and I've wondered this. Given the way that the anti-COVID vaccines have used mRNA against the COVID virus, would an approach like that work for universal flu, or what are some of the problems with that? Because, you know, I was involved with a universal flu project probably, you know, 8 or 9 years ago now, and it was one of the first things that I thought of when these guys actually use this really effectively for anti-COVID vaccines. So I imagine there have to be other challenges in thinking about how to do that.
There are so many different ways to getting to a universal flu vaccine. I mean, people are trying taking so many different approaches. And I'm sure there's going to be, you know, I mean, I'm not claiming that we're the only people who have the right idea. I do know that our idea will work because you know, the Vaccine Research Center at the NIH and Scripps together with Janssen, they both made headless molecules, and they made it so in these, you know, very complicated methods, and neither of them sort of like seem to be going forward. I think the one from the Vaccine Research is actually going into a proof of principle clinical trial, but nobody's picked it up. And so, you know, there's some problem with it. We don't really know what it is. Suffice it to say, though, that the level of protection that you get from these molecules is great. So, I mean, it really does work. We know the reason that Janssen dropped the collaboration with Scripps had to do with the stability of the molecule. So it really was that stability issue that we addressed with our technology. I don't know what the issue is with the Vaccine Research Center's headless molecule. There may be no issue with it. Maybe it's perfectly fine. I mean, it's going into the clinic. If they get a good result in the clinic, maybe they do license it. I have no idea. And there's a lot of other ways you could do it. You could do it with mRNAs. You know, Peter Palese had that approach where they were just swapping out different heads and sort of like, you know, and that seemed like a very good idea. It seems to me, though, that a lot of the approaches don't eliminate the head. And when the head is present, either in trans or, you know, sort of like, then you still have that immunodominant thing there. And as long as it's immunodominant and distracting from the stalk, it's not clear to me that you're really going to get a good stalk response. And that's sort of what. So we've decided that we're just going to take that clean approach where we just get rid of the head. And present only the stalk, and then the stalk antibodies would protect. So if you do an mRNA, you'll get very good responses, but you'll still have your head. So you can do 2 different or 5 different, or, you know, whatever, how many different sort of like, you know, HA proteins that you want to be working with, either in sequence or in parallel. I have no idea how people want to go about it, but we think our approach is great.
Right. So in thinking about how you've built the company, you've raised an exceptional amount of grant funding, right? The story is not the what seems to be now the tried and true version of, put together a PowerPoint deck, cash $50 million check from leading venture company, which if I read the press releases right, that only takes about 2 weeks. So you've gone about building things a little bit different. So you've taken you've raised an enormous amount of grant funding, and that's great in a lot of ways, but also I imagine pretty challenging. So what's it been like on the I don't know if roller coaster is the right way to characterize it, but what's it been like to build a company that way?
So so what the promise of an SBIR is that you get some cash with which you get to sort of try some things out and just get things working, so that by the time you bring in investment, that you've already de-risked some things, and that you've gotten to a place where the investment community will be more excited to get involved. The problem with grants is first of all, they're difficult to manage, and they're hard to get in the first place. Then they're difficult to manage, and then if you build a strategy on going from one grant to the next grant to the next grant, you have the risk of not getting it the first time, then you have the risk of not getting it the second time, and the risk of not getting it the third time,
and the gap between those is like 9 months to a year, right? It's not like you don't get it and then you just reapply,
right?
And it's not a short turnaround,
right? You have to have no, it's not. It's about a year. So you have to have a pretty good strategy. So when you apply for your phase 1, you apply for 2 years, but then you have to make sure that you can get it done within 1 year, so that after the first year, you're already applying for your phase 2, so that you have continuity. But then at the end of that phase 2, you still have to have the next phase 2 coming, and then you have to. You have to. We've actually managed to put together, I think, 10 years of grant funding, so the risk that you're going to fall apart at some point is enormous. And living with that risk constantly, just always constantly, I'm sure I'm not going to lead as long a life as I would have otherwise. It's definitely taken some years off of me on the end. It's incredibly stressful. And it just, a lot of times it just happened to work out, and it might not have, and so there's a lot of luck here as well. In 2013, we had to get a score. I think we had to get a score of 21 or something like that in order to, which is an incredibly good score for the SBIR.
I was going to say that's what, like 10th percentile, 9th percentile, something like that. That's incredible.
I don't know what the percentiles are, and the scores don't really translate to percentiles so nicely. We don't really get the information on how many grants were in that. We're down there, but suffice it to say, so the Division of AIDS probably made like 3 grants or something like that in that year, and we happened to be one of them. So because we got a 21 or a 20 or something, we were like one below. But like normally the cutoff is around 30 or 32. It's been in the last couple of years, but in the aftermath of the financial crisis, 2013 was a particularly tough year, and so we happened to get reviewers who really like it. But you just need one reviewer who has some, how it is with these reviewers. And
it doesn't even have to be a rational complaint.
And they're all really smart. They're really, really smart people. But if they have a perspective that if they're thinking about something else, then they're not as excited about what you're doing, and then they don't give you that amazing score. And if you don't get that amazing score in 2013, you just don't get funded. And there goes their company, really. So like, if that's how you built your strategy, which we did, we built the strategy on sort of like, we need to get to, we need to get very far along before we can get in front of investors. And that has a lot to do with the nature of the technology, because it's a process technology. So there really never is proof of principle of the technology until you've actually made it at scale in clinical manufacturing. So you have to actually get all the way there before you have proof of principle.
And that's just waving a red flag at reviewer number 2. That's really just begging for him or her to say, well, you don't have proof of principle yet. There's no way we could fund this. we actually had a really good question from the audience, because it's challenging to use grants to pay for everything, right? So the question was, one of the challenges that he's seen is paying for legal fees and actually paying, a CEO like salary, right? So there are all these kind of structural challenges to that as well.
Right. So no CEOs type salaries around here. So that's not how we've been getting through this. So we've held on to a lot of equity. We've gotten very far, but we've done that not with enormous salaries, but with just living frugally, and which has been a lot of fun. I really just being in control of my life, and sort of like just I don't have a boss really, so like my boss is the NIH, and the NIH is very forgiving. So that's been a very nice aspect of it. When it comes to the legal costs, so normal legal costs, when you're talking about just the cost of doing contracts or whatever, that's a fair and a valid indirect cost, and so you can cover that. When it comes to patent expenses, it's a different story, and so that's why I told you, sort of like that was one of the reasons why I needed to become a patent agent, to get through that, and reduce the costs of working with patent attorneys, because that is not a valid cost. So we still have the fee. So if you're running 2 or 3 phase 2s in parallel, you can actually you have some flexibility, right? It's 7%. All of our grants are always like $3 million grants over 3 years, so it's $1 million a year. So 7% of $1 million a year is on the order of $70,000. So if you have a couple of those going a year, you can get to a couple $100,000. So you can do some stuff with it.
We actually, and then we had a question, and you mentioned sort of that 1 million, the 3 million tranche with $1 million per year. Is that typical? Because the question was, what is the typical range of SBIR funds for a startup?
Yeah, so if you can focus your attention on focus areas or subject areas that are of particular interest to the NIH, which is not a bad idea, by the way, because that the NIH really knows where the hotspots are, and so like if the NIH is really focused on something, it's a good place to focus. So that was one of the reasons that we found RSV. Right, so we just knew that the NIH really liked RSV. So we was like, "What's RSV? How do you spell RSV?"
Alexa, how do you spell syncytial?
That's a hard one. I don't know.
Yeah, it is.
That's where you're thankful for autocorrect.
Yeah, but so if you stick to subject matters that the NIH is interested in, and they have these special subject matters or whatever, then you can get higher amounts than what are normally allowed. I think the normal amount is something, it's like $150,000 a year for 2 years for a phase 1, and then $750 for 2 years or something. But if you fall into these special categories, then you can get $300,000 a year in phase 1 and $1 million a year in phase 2 for 3 years. So that these are just so you just pick your subject matters. So if you have a technology and you want to apply it somewhere, apply it where the NIH cares about it, and you get more money.
And how do you figure out what they care about, right? Like, so how do you? Yeah.
There's lists. I mean, you go onto the SBIR website, or the special interests and sort of like all that stuff, and it's published.
Delineated.
Yes.
Yeah. The amounts are kind of funny though, because if you just go and read the stuff on the NIH website, it says very clearly that the phase 1 SBIR is this, and a phase 2 SBIR is this, but it's almost like the In-N-Out burger menu. Like if you talk to someone that knows all about SBIRs, it's like, yeah, but actually if you do this, you can get more, and you can push this into this year, and the whole bit. So there's a whole other thing that if you're really experienced on the grant side, the different angles. And honestly, the best advice I ever got with that is to talk to the program officers, tell them what you're doing, and really kind of start that dialogue, which is really kind of good advice for getting any kind of funding. You should have a dialogue with the people there.
Right.
Yeah.
We regularly get in the car and just drive down and give them presentations and make sure that, program officers are people, and they like to be taken seriously, and they should be taken seriously. They're really, really, really informed. They know their spaces really well. So if you go down and you give them a presentation of what it is that you think you should be doing, and they have some suggestions for you, those are usually really good suggestions. So we really make a habit of going down and talking to program officers. Now, during COVID, we couldn't do that, but we've done some Zooms. We like to be in touch and just get down there and talk to them.
Right.
We also have really good advisors. If I just may point that out. So there was early on in my SBIR writing career, I think I already had a phase 1. I got a cold call from this guy Neil Krieger, who was just recently passed away from COVID. But he gave me a call and just wanted to know whether he could help me writing grants. And we were, it was a cold call, and I was just like, I was on the phone and I was just like, why am I talking to you? Why am I talking to you? And we just kept talking and talking and talking. He kept on saying things, and just kept making me listen more and more. And he was very good at dropping names. And I really respect somebody who knows how to drop names.
Talent.
And anyway, so I ended up staying on the phone for I think 2 hours or something like that. And at the end, I was just like, so, but I just got a grant. Why do you think that I need to write a grant with you? I mean, I was just successful. So you're calling me for the very reason, I was on some list of grantees, right? That's what. So the very reason that you're calling me is the reason that I don't need to be working with you, so, or paying you. And he just sort of said, just give me a shot, just. And then he taught me how to write grants. Oh my God, he was so good at it. So having Neil teach me how to write grants, and just, get that structure down and really sort of be brutal and sort of the first couple of times he'd made me write 3-4 pages, and then he'd zoom in on that one sentence and he'd say, that's the sentence you like. Get rid of everything else you wrote. And so that's the type of, he was brutal with me, but we got very good at it. And it wasn't just, obviously I'm working with other people, and they're very.
So now you're making a transition that very few grant funded companies really ever get to make, right? So now you're actually looking at clinical development. You have the opportunity, I think, to go and raise private financing and stuff. So can you talk a little bit about that? What's it like talking to? So most of the time when people actually talk to investors, they're early. They usually don't have nearly as much proof of concept data. They certainly don't have as much what I would call scientific equity in the company, where you've actually kind of proved out the technology. So the venture community probably is justified in saying, well, this is what our kind of standard terms are. Is it a bit more challenging to kind of go to the venture community with more in your back pocket, because you probably don't look like you don't look like a typical company that they invest in. You're much further along.
Right. So that's both a positive and a negative, right? So investors, like to recognize patterns, and they're not recognizing a pattern here. So a lot of investors that we've spoken to say sort of, yeah, that's really interesting. And then it's just followed up by a long, yeah. So you get a lot of that. But then, you get, there are clearly some investors out there who are, as long as it's a numbers game, as I've been told, and so you find the guys who want to take a chance, and we've really made it very easy on them. So that was actually one of the questions to me. That was, so you're not a typical company. How do you deal with that? And the answer is, I can't deal with that because we're not a typical company. So I can't possibly make that go away. What I can do is take care of them on the back end, right? So, if you can get past, this non-pattern recognition and say sort of okay, we're different. We're not coming out of Harvard, and we're not, we came out of a management consultant who sat down and, with some friends, and dreamt up a technology and thought it would work and got it to work. You know, that's where we came from. So that's not, you cannot recognize that pattern. There is no pattern.
Right.
On the one hand. On the other hand, sort of like if you do get involved, sort of like these are all the things that we've taken care of. So there's a lot of upside to just getting past that.
Right.
And then sort of like in this particular round, we've focused on doing, so not only are we raising money, but we're licensing at the same time, right? So we're in conversations with a company in Asia that wants to license the technology for, you know, it's a limited license. We're not giving away the house, right? We're just giving away part of the company that we wouldn't otherwise be pursuing, really. So giving away that license gets us, first of all, it gets us cash. So there's, it gets us commercial, so they've done the diligence. So they're participating in the round, we get clinical data. Or at least I hope that's how it's going to work out, because, you know, if they're involved, you know, in the equity of the company, they should want to grow the company, and it really doesn't cost them anything to provide us the data,
right.
And we should provide our data to them. I really don't see any reason not to do that. We're in a partnership then, right? So doing a round of financing in combination with this license really takes a lot of risk off the table for
it does.
Right.
It's huge. And you know, the kind of corporate development and licensing angle on, you know, funding and company building, I think has become maybe slightly more mainstream. But when it comes down to it, you know, number one, you're right, you're taking risk off the table. You're accessing different kinds of capital too, right? And I think that because for a long time, one of the things that that venture folks would look for is your quote unquote, you know, validating partnership transaction, right? It's like, well, if you were to do a partnering transaction, we would be very excited about investing in this round, because that would mean that there's additional level of external validation of the technology. And now, you know, you now kind of do it in parallel, where the licensing group also says, yeah, we'd like to take a shot here too. And right. The nice thing that means is that it's now not all equity, right? You can actually put different kinds of capital into the company, and, you know, kind of not dilute yourself maybe quite so much as you would if it was just a straightforward equity round.
Right. And it gets us
out of curiosity.
So we go from manufacturing to phase 2 trials. That's a very nice position.
Yeah, no, that's exciting. So
in vaccines, you don't really sort of like, I mean, in our case, at least, I mean, I shouldn't go as broad as vaccines, but in our case, there really isn't a whole lot of risk in phase 1. So we really want to get to phase 2 as quickly as we possibly can, because that's where it starts to get really interesting, because that's where you start to divide between sort of like maternal vaccination versus the elderly, sort of like, you know, but how are we going to do this? And you have to start thinking forward, sort of like, you know, when somebody goes to the pharmacy and says, I would like to get an RSV vaccine, but I want the Calder vaccine. Sort of like what drives them to say that? And you've got to, and so you have to work back and sort of like, say sort of like, you know, what is it that we're trying to do here? And so, you know, and obviously we have a lot of ideas
Well, and in phase 2,
I was just gonna say, and then, I mean, to that piece, like different states have different requirements for who can vaccinate, and they may not go to a pharmacy. In some states, they may have to go to a physician, right? So,
yeah. I mean, on the bright side, we now, I would hope, have a general population that is slightly more educated about vaccines and perhaps vaccine choice, right? So there's at least hopefully you have that going for you. And at least from the phase 2 perspective, you have biology in phase 2, right? If you gave a bunch of healthy volunteers a vaccine in phase 1, you would get a safety profile, which hopefully would be perfectly fine, but everybody would expect it to be perfectly fine. So there's no, you know, there's nothing terrible. You know, congratulations, you did what we hoped you were going to do. Now do the hard part. You know?
Of course, we would also get we would get some data out of a phase 1 trial that would indicate sort of like it's actually working. Right? So, I mean, you don't when you're injecting people, you might as well draw some blood and look at their antibodies.
That's true.
It does drive the cost.
Although I can project the investor and reviewer to hand wave on, I'll believe it, you know, this would be really great to see in people that were sick, so I can see some outcomes data. So. It's
Oh,
easy to project the condescension.
I do think it's a good idea to include some older people in your first phase trial, right? So like, just include them, see what happens. Are you going to get over it? I mean, immunosenescence is a real thing,
Sure.
So
and
how do if your vaccine is so great, so like, how's it going to do in the elderly? Have a look at the antibodies in the elderly. That is definitely a plan. So for us, that's one of the main things that we want to drive, that we can overcome immunosenescence with great tolerability.
Well, that's another thing that I think that that COVID has really kind of highlighted, is that the either the elderly or the immunocompromised are a very real and patient set with a very high unmet need, right? These are the people that get the most complications out of these things, and we typically think about one set of patients with a particular therapeutic, but very often you have people with an immune system that's a little more frail or have outcomes that are much worse, and it's a real set of patients that we have to think about taking care of.
And then there's also cancer patients, cancer wards. I once spoke to the safety officer at Memorial Sloan Kettering, who pointed out that sort of like RSV outbreaks on cancer wards are a real issue. So there's a lot of places that this can play. It'll be fun to get in there and really start.
Yeah. So you mentioned your advisors before, and I wanted to ask you about this because you do have great advisors. I know Barry Buckland, I know Florian Chidel. They're fantastic. So how did have they been with the company for a long time? How did you actually get such a great slate of advisors? Because it's something that I think younger CEOs know that they kind of need to have, but most of the time aren't really sure about how to do it.
So I mean, I don't know how I did it really. I can give you a sequence.
You gotta tell
I think you should ask them what made them decide to sign on. I think if I go back, sort of like I think when the first place that we went from HIV was influenza. And there we started talking to Peter Palese. He came to IAVI and gave a talk. And so I that's when I suddenly understood. So I go, oh my God, the envelope protein, it's just a fusion protein. There's tons of these fusion proteins. Influenza has the HA protein. RSV has the F protein. So I went and I spoke to Peter Palese, and when I spoke to him, I realized that my friend Arthur, who you know very well, was working with Barry Buckland, and Barry Buckland was also working with Peter Palese through Protein Sciences. So Peter Palese was.
I think that's how we got to Peter Palese. Actually, I was at that company, and we were trying to do I think we were trying to do universal flu with Peter. So for those who don't know, Peter Palese is at Mount Sinai and is just basically a titan in the field of flu and immune responses, et cetera.
Right, and vaccines, flu vaccines. He's a virologist, but he also focuses on vaccines. Wonderful guy.
He is.
I was speaking to Peter, and we were talking about headless, and then we were I got in touch with Barry through Arthur, and then Barry just looked at the technology and what we were doing, and essentially agreed to watch. I think was the original sort of like, he was just gonna watch and see how this goes. And then as it progressed, sort of like he just got more and more excited, and sort of like, and then eventually someday I asked him whether he would join the scientific advisory board formally, and he did. So that's how we got him, or he got us, I think. In RSV, it was an even more convoluted story. So we were here at IAVI, and we realized that in order for us to apply the technology, we really need structure, because we need to engineer our tyrosine residues in very close proximity to each other in order for the dityrosine bonds to form. So we thought about sort of like, who are the structure people in HIV, and so we got in touch with Peter Kwong. And we started a collaboration with Peter Kwong at the NIH. And our go-to guy, the postdoc that we were working with in Peter Kwong's lab was Jason McClellan. And Jason McClellan was, this super bright, great, wonderful guy. And we loved working with him. And for a while there, he went quiet on us. And we couldn't really, we're just like, where's Jason? So like, we couldn't find him. And in the meantime, sort of like we were starting to think about RSV, and we got in touch with Barney Graham. And Barney was nice enough to invite us to the NIH to meet with him. So we went and met with Barney Graham at the NIH because we were going to be there anyway, because we were meeting with Peter Kwong. Jason McClellan joins the meeting, and we're just like, oh, what are you doing here? So like, and they had just solved the structure together of the RSVF protein. So that's when the RSVF protein, the pre-fusion conformation of the RSVF protein had been solved. And so that's how we just started a collaboration with Barney Graham and Jason to further stabilize the molecule. So they already had one version of a stabilized RSV pre-F, which is what they then licensed to GSK. And we decided to keep working and to stabilize that further. And so that's what we've done. And so then now Jason's on our scientific advisory board. And then when we had Barry and Jason, then we spoke to Florian, and then Florian joined. Yeah.
No, that's terrific. And it's one of these things that, I don't think there's necessarily a recipe for how to do it. And in general, sometimes the advice is, well, you want to build an advisory board, I don't know, call some people and figure out who you really want on there and why. So, and managing an SAB is an entire, it's a whole other, we don't have the time. It's a whole other hour to think about how to effectively manage an SAB. Bruce Booth had a great post on it probably a couple of years ago now. So you've now, now this journey has gotten to the point where you're hopefully, knock on wood, about to put some additional capital in the bank, and you've got to the point where you can get these things into clinical trials. So if you think back, what are some of the things that you wish you knew when you started?
Yeah. So when you don't have enough perspective, so you know how I just described sort of like, I really want to start thinking backwards. So when that person goes to the pharmacy and says he wants the Calder vaccine, that's where it all starts, right? That's where the money hits. That's where the rubber hits the road, right? So like, so you have to work all the way. You have to work from a scientist's perspective. You have to go all the way to the end, and then start working backwards, right? And you just don't have that perspective when you start out, and it's very hard to do. But the more perspective you can get on where you're trying to go, who's going to be thinking what, sort of like, how do I get into the head of an investor? Why are they going to say yes? Why are they going to take that second call? Sort of like, what are they, trying to think forward and sort of like trying to adjust your strategy and adjust what you're doing. That's just the most important thing you can do. And I don't think that I had that perspective at the beginning. That was the first thing. And then the second thing that I didn't realize is that this technology that I came up with, which, it was dityrosine bonding was described in the papers, and it's not like I came up with dityrosine bonding from scratch, but I decided to make a technology out of it. But what I didn't realize is that it's a process technology, and that you never get to proof of principle. There just is no proof of principle until you've actually made it and put it into people. And so that sort of like, that's really been that's why we needed to have so much SBIR funding in order to get to proof of principle, because we have to actually have a cell line and cross-link in a bioreactor, show that we can scale this thing, and that it's a lot of work that you have to do in order to get, and along the way, I always thought we had proof of principle. And then there was always sort of like, oh yeah, so like this isn't quite proof of principle. And then the next thing, and now why don't we buy a bioreactor? Oh, that's completely different. And so like, it just keeps going. So this concept of when you have proof of principle and how much you've actually de-risked. So like, that I wish I had thought about that, and I was a risk person. I came out of risk management, and I wish I would have thought about the risks.
If you described this to yourself, you would have never done it.
Correct.
It's really,
and I wouldn't do it over again. That's another.
the answer to quote unquote, when do you have proof of principle almost seems to be like soon, right? It's coming soon. We've just done this other thing. Yes, it's still coming soon. Just a little bit more, and just a little bit more. It's really, it's interesting. You do experiments and you advance the technology and you advance where you need to go, and then you realize there are yet more steps to the process that you're on. Although I would hopefully say that when you do actually go through clinical trials, I think the FDA authorization is a pretty clear demarcation of you can stop proof of principle now and you can actually go do the thing that you were trying to do.
Right. Although if you think about sort of like the mRNA vaccines, they didn't really have proof of principle until like 100 million people were vaccinated.
Yeah.
I mean, now they have proof of principle and for real, so, but I mean, it was, nobody really, I mean, at least I didn't think that it was going to be such a slam dunk.
I don't think anybody did.
It's a big surprise.
I don't think they did either. I think everyone,
who knows what they knew or what they thought.
But if you rewind, right, the general consensus around what we thought we were going to get or what we were hopeful to get out of the COVID vaccines was somewhere between 50 and 75% effectiveness, right? And none of the companies were really kind of banging the table and saying ours is going to be 95%. I think everybody saw the results and were pleasantly surprised that it worked as well as it did. So the other thing too, I mean, between both Moderna and BioNTech, the number of people that saw the actual scientific results prior to anything like this were few and far between, right? They had done a number of licensing deals. I imagine there's the investors that invested in them saw more than the general public did. But it's not like these were widely published things that everybody had a really good handle on. It really kind of an amazing circumstance that they got this opportunity to really kind of push everything into the middle of the table and have this kind of defining level of clinical proof of principle while everybody was watching. No pressure, guys, right? We have someone from the audience said, you just said you wouldn't do it over again, and then they said why, and then they said, well, it's possible that you may have been joking here. So I don't know. It's if you wrap everything together, do you wish you were still consulting?
No. I'm very happy with. I'm extremely happy with how things have gone. I had a really good time. The stress levels have been insane. And they're not going to get better. But sort of, would I do it again? Yeah, I don't know. Is the answer. That's a really hard, that's a hard question to answer. So like, would I do it again?
You mentioned this before, and
so I guess I was
just kind of
because I don't have an answer.
Well, I mean, you mentioned before basically doing consulting work for other companies and things, and it's something that I think most people don't really appreciate, that when you have something that's this up and down roller coaster, it's not like your salary is guaranteed every couple of weeks, right? And that is extremely hard to think about. Is it going to get better? Is there going to be more that comes in? And you end up doing these projects for different companies and stuff like that. It's disruptive. It's not terribly stable, and it really kind of eats on you behind the scenes. It's really hard.
And that's where we really need NewYorkBIO to step up and create more of, so because it's fine if there's a whole sort of like, if there's a whole, sort of like, if there's a whole community of biotech companies, which when we started, I mean, it's gotten really good now. But when we started, sort of like there was really, I mean, 98, there was not a lot of biotech in New York. There was some, but it wasn't a lot. So finding clients here to work for, so when there was no salary for a couple of years there, it wasn't that easy to find, sort of like the clients that you wanted to work with. Now you can just say, like, if my salary isn't coming from the company that I'm working on, you can just go, just go talk to somebody else and see whether you could fill in somewhere. And similarly, when my salary is covered and we have extra cash, we can bring in other people and people who are struggling right now with their companies, and sort of like they can do some work for us. That's worked really well. So,
I mean, for the record, Jennifer's only been here for 3 years, and it's been on a good uptick there. So
no. New York biotech scene is not comparable to what it was back then. I mean, it's really done. It's just.
And so we only have a few minutes left, but I did want to ask you, because you brought something up sort of in the New York biotech. Clearly I've been here 3 years, but and I think probably over the last decade, right? You've seen a real acceleration. How are you finding as you grow and build your company? Obviously, you're using somebody not in New York likely for your manufacturing, but how are you finding building your team in New York? Has that gotten easier over time as well?
I have to say, so like, we've never really had trouble attracting talent. The people that we've worked with have been just amazing. We're out here in the Brooklyn Army Terminal, which that's its own separate challenge. We're actually right now in conversations with Alexandria to see whether we could join or expand our space into the launch labs at Columbia. That might make things easier, and maybe we'll get even better people. But I can't imagine it because the people that we've been working with are just so good. So I don't think that's been a problem.
We actually hosted my very first New York Bio board meeting, right? Not like 6 weeks after I got there, in the IAVI offices in a conference room at the Brooklyn Army Terminal. It was very cool, but it took a very long time to drive there.
Yeah, the subway is faster, If you take this, it's on an express stop. The ferry stops right there. Yes. I mean, it's pretty easy to get to, actually. I also live in Brooklyn, so for
I was going to say, like, if we're couching things here, you're coming from Brooklyn. It's slightly different.
Right.
Yeah.
Getting to Columbia would be harder for me than getting here.
I was running that in my head when you were saying that. I'm like, that's really the reverse challenge. That's not difficult.
I'll be able to handle that.
The good news is we have so much life science real estate coming online, right? I mean, that part of the sort of problem of the equation is really being solved.
That must have been the main problem. I actually never dug into it, but sort of like, was that the main problem in New York? Sort of like that real estate was just didn't exist.
It was one of them.
It was
one of them.
Yeah.
It was a big, it's between lab space and capital. I think it's probably a chicken and egg scenario. But it's one of the main things that the EDC had a lever lean on. They did really well with it. A lot of credit to Alexandria for being first and making that really successful. And when you have that happen and other people start to come in, it's really a good snowball effect. I mean, to kind of think about where we are now, you've been at this for a long time, and I think there are others that have started companies a long time ago and have really seen the progress of New York. And now almost universally everyone is saying it's getting better, the environment is good, it's where people want to be. It has filled in the things that it was lacking as an ecosystem, and it's one of the reasons that we're really excited to be where we are, because we see these things popping up all over the place, and we just kind of know that the future is on a really big upswing. So
perfect.
Thank you. Listen, we can't thank you enough. We can't thank you enough for being here. I mean, you've got such a great story. Just the level of perseverance to bring things to where you brought them, and we celebrate every single time something that was grown here goes into clinical. So we're really excited for you, and kind of can't thank you enough for being here this morning and sharing your story with us.
All right. Thank you very much.
Thanks, Chris.
Right.
Everybody have a good day. And thanks to Moses and Singer for sponsoring.
All right. Thanks, everybody.
Thank you for tuning in to New York Bio's virtual breakfast series. Join us every Tuesday at 9 a.m. for more discussions with leaders from across the healthcare spectrum. For more information on New York Bio, please visit us at www.newyorkbio.org.